So in the last video, I talked about the signaling and the chemical synapse and how all the steps of that works together. Now we'll look at a few groupthink and then we'll continue on with our nerves lecture. So what would happen if neurotransmitter were not removed from the synaptic cleft? So neurotransmitter comes into the synaptic cleft, it binds to the ligand-gated channels, and that causes signaling to continue. What happens if the neurotransmitters are constantly there? So what happens is the ligand-gated channels would stay open, and eventually all the ions would balance between outside the cell and inside the cell, and there would be no difference between if there's an action potential or not, so signaling can't really happen. So what we need to do is find mechanisms to clean up the chemical synapse. And this happens through 4 really important processes. So the first one is called reuptake. So neurotransmitter that is in the synaptic cleft basically just gets recycled. It gets reuptake into the presynaptic neuron, and then it gets recycled and it's ready to be used again. Degradation. So neurotransmitter is broken down by enzymes in the synaptic cleft. So if you guys remember like acetylcholinesterase from the muscle cells, that was an example of a neurotransmitter being broken down by an enzyme. Third mechanism is diffusion. So there's no membrane holding the 2 neurons together, so a very small amount of the neurotransmitter can literally just be diffused. And then the 4th mechanism is absorbed by glial cells. So if you remember, like those astrocytes can absorb the neurotransmitter, and that will kind of take it out of the chemical synapse so that we don't have constant signaling. So based on the type of neurotransmitter present, that will trigger either an excitatory synapse, which is depolarization, or inhibitory synapse, which is hyperpolarization. So depending on the situation, the postsynaptic neuron can either be excited or inhibited. So in excitatory synapses, it's depolarized, and that moves it closer to threshold and more likely to continue signaling. Inhibitory synapses cause hyperpolarization, and they're further from threshold. So we abbreviate these EPSP and IPSP. So in an excitatory synapse that is closer to the threshold and more likely to trigger, what happens is the neurotransmitter binds to either a sodium or calcium channel. Sodium flows into the cell, makes it more positive. That causes depolarization, so it's closer to the threshold. Inhibitory synapses bind to either potassium or chloride channels. Either potassium exits the cell, taking its charge with it, or chloride enters the cell with its negative charge. That causes hyperpolarization, which makes it further from threshold and harder to fire another action potential. So after the action potential in one cell reaches the synaptic knob, there's a delay of about half to 1 millisecond before an action potential is generated in the postsynaptic cell. What would cause this? And would pathways with more synapses be faster or slower? So pause, write down your answer, and then continue to see the answer. So this delay is required for the synaptic signaling. So if you remember all those steps that happen, all that needs to happen in order to have signaling. And in general, the more synapses, the slower. So neurons can have multiple different pathways. There can be, for example, one neuron that splits into many, or there can be many neurons that converge into one. So convergence, one cell is influenced by many other. So here you can see there's one neuron and there are all these other neurons converging together to make one. Divergence, so here this one neuron is splitting up, it's dividing into signal many other neurons. So we're going to talk about neurotransmitters and different classes of them. Don't panic, you don't need to know all of them, but you do need to know the stuff in black. Okay? So neurotransmitters, like we said, are small chemical messengers that quickly get removed from the synaptic cleft. And this is what two neurons use to talk to each other. So neurotransmitters can be always excitatory, always inhibitory, or they can be either, depending on the situation. So if they're excitatory, they are causing depolarization. If they are inhibitory, they are causing hyperpolarization. Neurotransmitters can be fast, that work through those chemically gated channels or ligand gated channels, or they can be slow, which work through a secondary messenger pathway. And we call these neuromodulators, and I'll go through what a second messenger is in a second. So all the neurotransmitters get classified into one of 5 main categories. The first category is acetylcholine. Acetylcholine is this molecule here. I don't care that you know the drawing of it or anything, but just know that that is so important that it gets its own category. Next category are amino acids. So these are the building blocks of your proteins, and they can be used to signal as well. Monoamines, these are the ones that you guys have probably heard of. So you guys have probably heard of catecholamines, like epinephrine, norepinephrine, and dopamine, or indolamines, which are serotonin and histamine. The fourth category are called neuropeptides, and they usually act as neuromodulators that are a little bit slow working through that secondary pathway that I'll explain in a second. Examples, substance P, which causes pain in the body, or opioids, you guys have probably heard of. An example of that are your endorphins, enkephalins, CCK, morphine that's used in the hospital as a painkiller. The fifth category are dissolved gases, also usually neuromodulators. Example is nitric oxide that's used in surgery. The sixth category, everything else. These are in red, don't worry about them. So just kind of keep it in mind, like ATP can be used as a neurotransmitter, insulin, glucagon, like all these weird things can also be used as neurotransmitters. Neuromodulators act via a secondary messenger system. This causes long-term changes in the synapses. So a secondary messenger system is very complicated. You can look it up or email me if you have questions about it. But basically what I want you to know is that a second messenger system is a really lengthy chain of different steps that causes long-term depression or enhancement. It leads to changes in DNA and changes in the number of proteins produced. So for example, a second messenger could cause an increase in sodium ligand-gated channels. So that would long-term make the cell easier to depolarize, so it would enhance the synapse. Some examples, neuropeptides and nitrous oxide here. So neuropeptides are just chains of amino acids, and they have longer lasting effects. I don't really care that you know all the specific details about these. So in our basic drawing of nervous system signaling, we kind of just said, well, it kind of depends on what type of channel protein is present that will cause either an action potential or inhibit it. Here we'll look at 3 specific types and what they lead to. So the first one is an excitatory cholinergic synapse. So it is excitatory, so that means that it's going to increase the likelihood of action potentials happening. It's cholinergic, so acetylcholine. Okay. So what happens here, the nerve signal causes the calcium channels to open, acetylcholine gets released. When acetylcholine binds to the postsynaptic neuron, that causes sodium channels to open, and that causes depolarization and a local potential. If the postsynaptic cell reaches threshold, an action potential is triggered. Okay, so the release of acetylcholine causes sodium channels to open, which causes a local potential. The second type of signaling here is called an inhibitory GABAergic synapse. So here, same steps happen, but the neurotransmitter that's used is GABA. When GABA binds to the postsynaptic neuron, chloride channels open and chloride goes into the cells. Chloride has a negative charge with it, so it causes hyperpolarization in the neuron. So it is now less likely to reach threshold, so it is inhibitory. Another example is the excitatory adrenergic synapse. So here, the neurotransmitter that gets released is norepinephrine. You'll see it abbreviated NE. Norepinephrine works through a secondary messenger system, and I don't care that you know all the specific details, but basically it eventually causes a release of cyclic AMP, which can synthesize new enzymes. So for example, it could synthesize more enzymes that degrade the neurotransmitter in the synaptic cleft. If you guys remember that, acetylcholinesterase would be an example of that. So CAMP could synthesize more acetylcholinesterase. This slide is just showing a detailed schematic of a secondary messenger system. As you can see, there are a lot of different steps going on here. I don't care that you know all the details of this. This is just kind of FYI. If you're interested, feel free to email me and we can chat about it. So again, we need some way to stop signaling from happening. So we have three ways of doing this. The first is diffusion. So the neurotransmitter goes away from the synapse. The second is reabsorption. And then the third step is degradation. Okay, so acetylcholinesterase degrades acetylcholine. With the reabsorption here, there's a really important enzyme called monoamine oxidase that breaks up the monoamines. You guys have probably heard of MAOI inhibitors like Prozac, and we'll talk about that on the next lecture. Okay, so look at these questions, and then we'll go through the answers on the next video.